She actually runs the graduate program as part of being the vice chair. And she's in charge of the whole graduate program, she by herself, not a committee, anything, she, of the whole graduate program in the department. And she has been doing that since her second year as assistant professor, but officially as vice chair since getting her tenure. I only have really good words to the work that Joanna has done over these years as a mentor, as a supporter of junior faculty, as reviewer in very many panels in NSF. And it's really a pleasure to have you here and sharing your new work, or at least the latest that I read. I'm just going to tell you two little things. It's all about post-translational modifications, in particular glycosylation of proteins, and how the oscillation of the oscillatory pattern that she sees and glycosylation relates to circadian physiology. And she really points, or she's going to really point to nutrients as input for the circadian clock. So without further ado, Joanna, the audience is yours.
All right. Can you hear me? All right. I'm so used to asking that because this is actually the first talk I've given since the pandemic began in person. So yeah. Forgive me if I'm a little rusty. Okay, so thank you for inviting me, Kala, and thank you for introduction. It's great to be here to see some of my friends and meet new people. So as Kala mentioned, so my lab works on regulation and function of biological rhythms. I think since my postdoc, I've been working on molecular mechanisms that regulate circadian rhythms, which is the 24-hour rhythms. But the last, I think, 4 or 5 years, we've started to actually look at seasonal rhythms as well. And in particular, we're interested in actually understanding how the circadian clock can inform calendar year, because as you can imagine, in the summertime, day length is longer, wintertime, day length is shorter. So how those two rhythms actually connect to each other is not clearly known. So we've been working on that as well. But for today, I'm going to focus on just circadian rhythms. If you look at all the work in my lab, you can probably group them into 3 different groups of questions. Beyond just the fact that I also work on pest management, just that's what the strawberry and cherry grants are for. So that's totally different. So if you look at just the work in biological rhythms, you can group them into 3 different groups of questions. The first group of questions is we try to study the proteins and the genes that actually make up the core of the timers and the clocks and how they work together to keep time. The second group of questions, it's about how these clocks and timers can integrate environmental as well as sort of metabolic signals to keep time, to entrain to local environment. And then finally, we also ask questions about how these clocks and timers regulate output rhythms. And what happens when these rhythms are disrupted, what's the influence on health and fitness, et cetera, et cetera. So for today, we're going to focus on a particular question, which is how animals can actually use nutrients and metabolic input to keep time. It's a little bit different than a lot of what a lot of people have studied in the past decades, and in terms of how light and temperature, which are common signals for the clocks, how that is leveraged by the clock to keep time. We're asking how the animals can actually use metabolic input and food as time cues. So this, hopefully by the end of the talk, it will sort of causes you guys to think about, when you're thinking about eating healthy, not just what you're eating, but also the time of the day when you actually consume food. This is not something that a lot of us actually thought about, at least for myself. I know before I started work on this, I used to eat really late, and that is actually not very healthy. And hopefully you'll get that at the end of the talk. And perhaps rethink your midnight snack, which I now don't do anymore. And as students, you can also use it as an excuse to tell your PI why you need to actually start your happy hour in the middle of the day on Friday. So yeah, not start at 6 or 7. because beer counts as well as food. It's carbohydrates. Okay. Before I get to the actual science and the questions, I just want to give those of you who might not be as familiar with clocks and timers a little bit of background on circadian rhythms. The first thing I want to tell you is why we actually need clocks, circadian clocks, and why it's especially important. You can probably group them into three main advantages. The first one is we live in an environment that have very predictable day-night cycle. Accompanying that, it's actually very predictable changes in the environment. So it would be really great if you have an endogenous timers that you can use to predict those changes. So instead of just reacting to those changes in the environment, you can actually anticipate them. So that's a really good advantage of actually having an endogenous timer, right? The second advantage that having a clock would give you is that it allows you to actually use the resources in the environment, which are often limited, use them at the most biologically advantageous time of the day, right? So this is an example given by Michael Young, who's of course won the Nobel Prize for studying circadian rhythms using Drosophila as a model in 2017. So he gave this example in his Nobel lecture. These are actually pictures that he took when he go on a hike. I don't know where, but he encountered this particular plant at 2 p.m. on the hike out on the trail. And you can see that these flowers, the petals are not open yet, right? But then what happens is on the return trip at around 6 p.m., you can see that the flowers are now open. Something wrong with this picture. Anyway, so I think there was supposed to be a picture here that says the flowers are open. So just believe that they're open. So this plant lives in a desert environment. So there is not a lot of water in the environment. In order for them to actually open the petals, they need to actually use water. So they need to pick the right time of the day in order to do that. The pollinators of this plant is actually hawk moth, which actually is active in the nighttime. So it doesn't make sense for them to actually open their petals all day long. So they only choose to actually do that just before the pollinators become active. So this the clock allows them to be able to do that. So it's really important for efficient use of limited resources. And the last advantage that I usually tell my students about having a clock, it's about really synchronization of different organ systems and different tissues and different cellular mechanisms in your body. If you can imagine your body is sort of analogous to a factory where there's different machines and assembly lines and they all work together, right? So if they are all on the same time schedule, they will be more efficient and work better together. So you can imagine that the circadian clock also enable temporal synchronization of all your physiological system. But it's not just at an individual level. You can imagine that if you're in a population, if you're totally out of sync with other individuals in the environment, you might actually not survive very well. And in the next few slides, I'm actually going to give you an example in mosquitoes, which actually speak to that. So with all these different advantages, it's not surprising that wherever scientists look, most organisms from bacteria all the way to humans have some kind of an endogenous clock. They might not all be using the same clock genes or clock proteins, but they do all have some kind of a timing mechanism because having a clock has such advantages. So just to expand that idea a little bit, not just talking about the clock itself, the clock is actually part of a circadian timing system, which not only include just the timers, which is sort of at the center of the circadian timing system. And there's not just only one clock, as many of you know, there's clocks in the brain, there's actually clocks all over your body in your individual cells, and they all have to be in synchrony in order for the whole system to work well. They get their timing signal from input pathways that senses different time cues, including, as I mentioned before, light, temperature, but also things like food, which we're going to focus on later on when I talk about the science, as well as like something like weird, like vibration. And a lot of people are starting to actually look at exercise as a time cue as well. And so all these different inputs, and of course, there are many more that probably we don't know about, provide the time cues for the clocks to entrain to their local environment. And this in turn allows the clock to regulate all the different outputs of the circadian clock in different aspects in your physiology, metabolism, behavior. And down here, these are just some examples of circadian outputs that a lot of people, a lot of scientists have been studying, including sleep-wake cycle, but also immune response, et cetera, et cetera. And because I'm from the Department of Entomology, I want to specifically point out 2 examples in insects. I don't know, I met a couple of mosquito PIs early on, so I'm assuming they're insect people here. So one really famous example is the clock it's important for something called time compensated sun compass that is used for navigation, long distance navigation by the monarch butterfly. So if you can see here, this is a butterfly that actually have a functional clock, and it uses this clock to understand which direction it should fly to for migration. So when it's flying south at 10 a.m., 12 p.m., and then 2 p.m., you can see that it uses the sun as a reference and actually travel to different directions. If you have a butterfly that actually don't have a functional clock, it actually is going to just fly towards the sun, which makes them get lost. So the clock is really important for this specific aspect of monarch butterfly biology. This is another example of circadian output and the function of the clock. This is a paper published in Science in 2021, showing that mosquitoes actually, it's important for regulate timing of pheromone synthesis, swarming, and mating. So this is also very important work. It illustrates the fact that it's not just synchronization at the individual level between different organs, but also between different members of the whole community, so at the population level. So now that we understand sort of the circadian timing system and its function, so I want to point out to you that, which is relevant to what I'm going to talk about, some of the output of the circadian clock, especially what I want to focus on today is feeding fasting cycle, also feeds back to actually increase the robustness of circadian physiology, sometimes by actually feeding back to and regulate the function of the circadian clock proteins in the core clock, but also in other cases, just amplifying circadian physiology at the level of, post-transcriptional and post-translational level. And all the system creates is like really robust physiology. So we're interested in particular in the timing of feeding fasting cycle and how that feeds back to the clock, because not only because we're interested in understanding the basic mechanisms, but because they're also like clinical relevance to the whole questions. So there has been tons of work done on time restricted eating or feeding, depending on what organisms you're talking about. Done by mostly Sachin Panda's lab in Salk Institute in San Diego, but also the late Sazon Kossi's lab. And so they find that time-restricted eating has actually a lot of protective effects against metabolic diseases and obesity, et cetera, et cetera. And time-restricted feeding or eating, for those of you who are not familiar, it's just basically a discrete windows of food availability. For example, like for me, after I started working on this, I don't eat after 7 p.m. Usually after 7 p.m. I give excuses to myself. I do that over the weekend just to give myself a break so that I can, go to a happy hour later. But so time restricted feeding actually have really good metabolic influence. And it actually helps to reduce insulin resistance, increase glucose tolerance. And there's also a lot of other influence and beneficial influence as well. So this is why, also one of the reasons why we're interested in studying this question.
There's not much on losing weight, right? It's more about being metabolically
healthy, right? Exactly. And in fact, I think most of the study that it's done by Sachin's lab, it's really on health effects when you consume fatty diet. For more normal diet, there's less of the benefits, but I think more needs to be looked into that. But I think so far, most of the data has been on people who are consuming high fat diet or actually have metabolic diseases like diabetes, et cetera, et cetera. Yeah. So, like I said, we're interested in the mechanisms that mediate the feedback by the feeding fasting cycle, which is essentially metabolic input into regulating circadian physiology. We're not the first person or the first group that actually have been looking into this. Over the past, I think, decade at least, there have been many groups interested in this. And this is one example. For example, if you have fasting, we know that that actually changes AMP and over ATP ratio, and that actually influence and modulate AMP kinase activity, which then phosphorylate cryptochrome 1, which is an important clock protein, leading to its degradation. And that's how feeding and fasting can actually feed into the function of the core clock. This is another example where feeding can actually increase the activity of all these metabolically sensitive kinases like TORC1, AKT kinase, SXK kinase, and that leads to increased phosphorylation of a lot of proteins in our body. And the end result of that is actually increased protein synthesis, which makes total sense. These are just two examples, and there are a lot of other examples reviewed in, very nice reviews people have written. This is one particular one that I like from Sachin's, that he talks about all these different metabolites, energy status that can actually influence circadian physiology as a feedback from feeding fasting cycle. So for us, the last, I would say maybe 5 years or so, we have been studying this particular mechanism as a way for metabolic input to feedback and regulate circadian physiology. So we're interested in this particular post-translational modification called O-link glycosylation. It's just addition of sugar group, a particular sugar group onto proteins that changes the protein structure and function. And the reason why we're interested in this, I would say there are 3 main reasons. One is since I've been a postdoc, I've always have been interested in understanding how phosphorylation regulates circadian physiology, as Kala mentioned. It's interesting that O-link glycosylation, just like phosphorylation, also targets serine and threonine. And in the past decade, it's been shown that there's actually a lot of interplay and interaction between these two post-translational modification, phosphorylation and glycosylation. So that is sort of what draws me into this. Before then, I was trying to avoid metabolic regulation because so many people are working on it, but it just came to a point that I cannot avoid it anymore. So I have to kind of jump in. So that's one reason why we're studying O-link glycosylation. The second reason why we're studying O-link glycosylation is because we find that this O-link glycosylation is actually the product of this particular pathway, hexosamine biosynthetic pathway. The end product of this pathway, also called HBP, is actually UTP-glycinate. This is the sugar group that is the donor for this modification. And because we're interested in metabolic regulation, and this particular modification is so sensitive to nutrient, we decided that, like I said, we cannot avoid it. So we started studying this as well. And then finally, we have this idea, this hypothesis that perhaps because there is such interaction between phosphorylation and glycosylation and phosphorylation regulates all kinds of metabolic, all kinds of physiological pathways in the body, perhaps changes in glycosylation can actually modulate and changes the whole phosphoproteome. So this is a very interesting question to us. And that's why we decided to study like this particular pathway with regard to how metabolic input regulates circadian physiology. So to start addressing this particular area, we asked first 3 basic questions. So very simple. Does O-link glycosylation of cellular proteins just globally exhibit daily rhythmicity? So before this, I didn't tell you that we have a paper in 2019 in PLOS Genetics showing that some of the clock proteins are actually regulated by glycosylation, and the modification actually exhibit daily rhythmicity. So we want to extend that to see whether other cellular proteins beyond just clock proteins also exhibit daily rhythmicity in this particular modification. So that's the first question. The second question is daily glycosylation rhythm sensitive to feeding time, right? That's actually kind of the key question, which we really want to know, like that you eating at nighttime versus daytime, does that actually changes the modification? And does that leads to changes in protein structure and function, right? That would means, that it's really interesting when you actually eat at night, your proteins are actually modified differently. So that was actually pretty scary when we first start working on that. Every single time you pick up food, you're like, how are my proteins getting modified? But anyway, that's the problem of biochemists, I guess. Finally, the question is, does the clock regulate this rhythmicity besides dictating feeding fasting cycles? Start waving your arms. Thank you. Yes. So does the clock regulate this rhythmicity besides regulating feeding fasting cycle, right? Because we know feeding fasting cycle is one of the main circadian output of the clock. But besides that are, for example, HBP enzymes also regulated by the clock. Are there different levels of regulation? So before I get into telling you the answers to these questions, I wanted just to mention that all this work is done by one grad student that I'm going to tell you about. It's Nitro. She actually graduated last year, 2 years ago. I don't remember. But anyways, the reason why I don't remember is because she's still in the lab. So she's now a postdoc in the lab trying to move this into the mouse model. And at the end of the talk, I'll give you some new data that she has, which is in the mouse model. But what I'm going to show you in the first part of the talk, it's all done in the fly model. Okay. So let's start with the first question, which is, does glycosylation of cellular proteins actually exhibit daily rhythmicity? So to do that, we had previously optimized a chemoenzymatic labeling approach to actually tag glycosylated protein with biotin. So this is shown in panel A right here. And you can actually use Olin glycosylation antibodies. They're just in combination with Western blot. We tried that when we first started the project, but we find that the antibody is actually really non-specific. So we then actually use this particular approach, which is shown here. If you tag glycosylated protein with biotin, you can actually use streptavidin to detect it. So it's really nice. And it's very specific, much more sensitive. So what I'm going to show you today for the rest of the talk, it's a lot of results based on this particular technique. Okay. So just to verify it and validate this approach, we can actually overexpress the enzyme oligate transferase in flies and actually see if that actually increase Olin glycosylation. And we use PER as sort of a test protein to show you that this works. So when you actually have wild type flies versus OGT overexpressive flies, you can see that Olin glycosylated PER is actually increased when you have overexpress of the enzymes that add the sugar group onto the protein PER. And you can actually do the reverse where you do OGT RNAi, and you can actually see a decrease in the signal on the different clock proteins. So this technique actually works beautifully. So we started using this to look at global rhythm in, daily rhythm in global glycosylation is just basically protein extract. So you'll see a lot of these graphs right here where the x-axis is just time, and most of them are double plotted just to show you the rhythm more nicely. And then the x-axis is just signal intensity of the glycosylation level. And for those of you who are not familiar, CT just means Zeitgeber. Zeitgeber is like a time giver. So CT0 in our case is when the lights turns on. CT12 is when the lights turn off. So you have a 24-hour day, right? So as you can see in panel A, in the top panel, you can see glycosylated protein. And we actually extracted nuclear proteins and we label with biotin all those proteins that are glycosylated. And we just use we use streptavidin to actually detect biotin in combination with Western blotting. And you can see that total protein is for loading. It's clear just in the quantification of the results that you have a really nice circadian daily rhythm, not circadian rhythm, but daily rhythm. So we're really excited about this because it's not just clock proteins that are exhibiting rhythms in glycosylation, but also just sort of globally in nuclear proteins. And you can imagine that, this rhythm could be attributed to the strong feeding fasting cycle in the fly, right? In the past, people have shown that flies, just like humans, exhibit feeding fasting cycle, but we just want to make sure that that's the case. And in flies is really easy. We just use a cafe assay. The name is very appropriate to actually feed the flies continuously, and we measure how much they're eating and consuming. And we use this approach to show that, yeah, indeed, like what people have done before, you get a really nice feeding and fasting cycle. And that translates to actually rhythms and daily rhythms and very strong rhythms in metabolites. So this is in panel B and C. This is GCMS metabolomics showing that over 4 time points over a circadian day in both bodies and heads, you get nice rhythms in metabolites, especially in the body, which is not surprising because it's more metabolically sensitive without the blood-brain barrier. And this is just showing that the percentage of rhythmic metabolites is much higher in the body versus heads, which is, again, not surprising. Because we're interested in Olin glycosylation, and that's it depends on HBP, the hexosamine biosynthetic pathway, we look further into specifically HBP metabolites. In particular, you don't have to look at all of them, but just focus sort of the end product of the HBP, which is the UDP glitinac, the donor molecule for the modification, the sugar modification. And you can see that it actually really nicely have a daily rhythm, especially in the body, which is the line shown in blue. In the head, you see sort of a dampened rhythm as well, but it's still rhythmic. So this means that it's plausible that the clock regulates feeding fasting cycle, which then translate into metabolic rhythms, which then translate into sort of this global and specific rhythm in glycosylation in proteins. But all this is correlative, so we have to actually show causation. So what we do is, and to answer the second question, we actually manipulate feeding time, which is pretty easy in flies, right? We just don't give them the food until we want to. Okay, so this is the experimental scheme that we have that Nitro has. So at lib is just like, for those of you who work on mice, it's just you leave the food out for the mice. They eat whenever they want, even though they mostly eat, in a rhythmic manner, but there is some occasional snacking. That's basically the data I showed you in the previous few slides. We also did restrictive feeding. So based on a lib feeding assay, we know that when they prefer to eat. So we actually do a ZT21-3 restricted feeding. In flies, they tend to eat, at close to dawn. So what happens is we actually limit a 6-hour feeding window close to dawn, ZT21-3. But we also have what we call unnatural feeding time or restrictive feeding at CT9-15. So kind of like an anti-phase to the natural feeding time. And then we just chop up the flies, get the bodies because it's more metabolically sensitive. And then we detect a level of glycosylated proteins like globally. So when you look at glycosylated rhythm in these 3 different fly groups, the first one in red is the at lib, which is sort of what I already showed you. There is some rhythm that is based on circadian statistics. It's rhythmic. So it's nicely rhythmic. And the glycosylation is highest close to dawn. And then the blue line, it's natural feeding time. You can see that you still see that nice rhythm. Maybe the amplitude is even slightly higher based on circadian stats. That's the case. And then with the yellow line, which I'm going to show you next, this is for unnatural feeding time. So when you shift to nighttime feeding for flies, so imagine if you're eating at night, what's going to happen, right? So originally, Nitro thought, oh, maybe the peak will shift, but that's not the case. So it's actually flat. So the rhythm is totally dampened. You don't get a phase shift instead. And this is not because the flies are eating less. right? Because you can imagine it's possible that these flies are eating less, so you have actually less metabolic input and consequently less glycosylation. But that's actually not the case. So we measure food consumption. This is in panel B. You can see that the unnatural feeding group actually eats more. And this also happens in humans. Like when you eat at night, you tend to eat more. And I can personally say that that's true because we experience it. And so that's not the case. It's not dampened because they eat less. They actually eat more. And in C, we're actually also measuring food consumption, but only at the time window when we have the specific feeding for the natural and the feeding and the unnatural feeding group. In both cases, you can see that in the unnatural feeding group and the natural feeding group, they both eat more and then they ad lib. So in this case, again, this really shows you that the dampened rhythm, it's not because the flies are eating less, it's just something else is happening, right? So we think that based on this data, the dampened glycosylation rhythm in unnatural feeding, which is the yellow line, suggests that there's some kind of clock control gating mechanisms that is normally preventing high glycosylation level at non-optimal time, right? So this gets to the last question, this set of questions is, does the clock regulate this rhythmicity besides dictating feeding fasting cycle? There must be something, some kind of clock gating mechanisms in addition to regulation of feeding fasting. So what is it? So the first key experiment that we did was, since we have a lot of clock mutants, what we did was we just used one of the arrhythmic mutant per 0, and we control feeding, only feed them at natural feeding time to take sort of the clock regulated feeding out of the question, and we see what happens to the glycosylation rhythm, right? So you can see that in panel A, the black line is wild type flies, and we're feeding them at only natural feeding time. And then per 0 flies, it's a arrhythmic mutant. We're also feeding it at only natural feeding time. And even when we do that, you can see that there is that dampened rhythm again. So it must be regulating something else in addition to regulating feeding fasting cycle. And again, the control here shows that it's not because they eat less. Okay, so what is that clock gated mechanism or mechanisms, right? So one thing that we started looking at is the clock regulating some of those enzymes in the HBP pathway that produce that sugar molecule UDP GLITNAC, right? So that's the first thing we looked at. So the way that we try to look at that and see which is the enzyme in the HBP pathway, which is shown here on the left, is regulated by the clock, is by actually doing targeted metabolomics to look at metabolites. The idea is that if there's a specific enzyme that is regulated by the clock, if we change the feeding time, maybe we'll see the metabolites accumulate abnormally. Okay, so the red line is from flies that actually are eating at natural feeding time, and the blue is from unnatural. And we're measuring two key metabolites here, glucose 6-phosphate and fructose 6-phosphate. We measure all the other ones as well, but these are just the two that I show you because they're interesting. So you can see that the blue line and the red line are what I just told you. And there's these blue boxes shaded and the red boxes. These are the time when they actually were consuming food. So just to match them and so that you can see the results easily. You can see that in both of these metabolites, it seems that in the unnatural feeding group, these metabolites actually accumulate right after feeding. It goes abnormally high. This one over here and this one over here. And this would make sense if the enzyme that is right immediately downstream to it, it's actually regulated by the clock. And they're simply not active during this time of the day, right? That would make these metabolites accumulate. And that's what we think. And based on this result, we think that GFAT, this is the enzyme that might be actually be regulating by the clock, and that's preventing these metabolites from being metabolized. So it's possible that the clock may regulate GFAT mRNA at the transcriptional level or at the enzymatic activity level, right? So we looked at both. but before we do that, we just want to make sure, even though it's pretty expected, that GFAT is an enzyme that is important for protein glycosylation. It has to be, we think, because it's a key enzyme in the HBP, but we still have to test it. And it's pretty easy to do that in flies because there are mutants. So we have a GFAT2 mutant. GFAT2 is the main GFAT in the body of flies that are active. And when we actually look at GFAT2 mutants, we show that in the glycosylation is actually dampened. So it's definitely needed for protein glycosylation to actually peak at the right time. And this is just a graph, a control showing that the GFAT2 mutant that we have actually have decreased enzymatic activity. So now that we've shown that, the first thing we want to look at is GFAT enzyme activity regulated by the clock. So we have three groups of flies here in this graph. One is wild type flies that are fed at natural feeding time. Wild type flies that are fed at unnatural feeding time. This is in blue. And then per-zero flies that are fed at natural feeding time. So again, you take that out of feeding out of the equation, but you knock down the clock. Okay, as you can see here, again, the feeding window is also shaded according to the line, the color of the line. So you can see that GFAT enzyme activity, it's rhythmic according to circadian statistics. We actually calculated that. In wild type flies, feed at natural feeding time. The blue line, which is the unnatural feeding time, it's still kind of rhythmic, but it's way more dampened. And in per-zero, it's just all over the place, which is not surprising because if it's regulated by the clock, GFAT enzyme activity would be all over the place, right? Even though it's actually, you control the feeding time. Okay, so from this graph, we see that yes, GFAT enzyme activity, it's regulated by the clock somehow. We don't know how, at what level, but we know that the enzyme activity is actually rhythmic. Based on, unfortunately, we don't have a GFAT antibody, and the reviewers didn't ask for it, which is great. So we concluded that GFAT, probably the enzyme activity, is probably regulated by clock in some post-translational regulatory mechanism, presumably GFAT phosphorylation, because it's been shown in mouse in a phosphoproteomics data set to be rhythmic in mouse liver. So we kind of like just borrow that. On the other hand, it's also possible that GFAT enzyme activity is regulated rhythmically at the transcriptional level as well, right? Maybe GFAT2 mRNA, it's rhythmic. So we tested that as well, but that doesn't seem to be the case. So here, the coloring is the same as this panel A right here. We look at GFAT2 mRNA by qPCR, and interestingly, GFAT2 mRNA is actually induced by feeding. It's not regulated by the clock as well. So you can see that because the GFAT2 mRNA peak every single time, depending on the feeding time, right after feeding, even in per-zero. So this gray line right here, you can see that it peaks right after the pink window, which is when it's finished feeding. So we think that based on these two pieces of data, the coincidence of clock-regulated post-translational modification of GFAT, possibly by phosphorylation, and the feeding-induced elevation in GFAT mRNA in natural feeding condition actually enhances HBP output and Olin glycosylation rhythm. And this can explain why this blue line right here is slightly dampened, the enzyme activity, because at this same time, you can see that the mRNA is just less, meaning that will be less protein. And then this is why the enzyme activity level is actually lower. Okay. So we think that GFAT2, the enzyme is actually a key contributor to the daily rhythmicity in glycosylation. But then there's also other factors that might be involved. And we think that OGT and OGA is also involved, and we tested that, and that turns out to be true. These two enzymes are also regulated by both the clock as well as feeding. Just for the interest of time, I'm going to not show you that data, but it's actually published in Nitro's paper in 2021. So if you're interested, you can go take a look. And at the end of all that work, so Nitro combined all that work and formulated sort of this model to show that GFAT, OGT, OGA are all key points for integration of circadian and metabolic signals to regulate daily rhythmicity in Olin glycosylation. And we think that that's actually contributing to rhythmic biology, circadian physiology. So just to summarize what I've told you, Olin glycosylation of nuclear proteins exhibit daily rhythmicity in flies. We actually did not look at cytoplasmic protein in this particular paper, and we're definitely looking at them because there are a lot of other proteins that might be interesting and they're in the cytoplasm, right? So that would be next. We also find that daily Olin glycosylation rhythm dampens if animals are fed at unnatural time. For example, if you consume food at nighttime, because there's simply a conflict between light dark and also just the metabolic time cues, which is never good. And then finally, the last 2 points, the circadian clock regulates not just feeding fasting rhythm, but also key enzymes that contribute to the daily rhythmicity in protein Olin glycosylation. Okay, so what are we doing now? Like, what are the studies that we're doing after that paper? So we wanted to do two key things, but of course there's always other things that we're doing. But the 2 key things is we want to identify the proteins that are regulated by Olin glycosylation, especially for me, like what are those proteins that are regulated by the interplay between phosphorylation and glycosylation, and what are they doing in terms of circadian physiology, right? The other question that mostly Nitro is really interested is how whether different organs, in different organs, circadian and metabolic signals get integrated differently. Because you can imagine for the heart versus the liver, the integration, the input from the brain clock and also from metabolic input might be different. So it's interesting to actually look at different organs. But as you can imagine, it's going to be really difficult to actually study that in flies because they're itty bitty. It's really difficult to actually get enough tissues to do proteomics and metabolomics to dissect, little hearts from the flies. So we're turning into the mouse model to do some of this work because I find that the mouse liver is actually much bigger. Yeah. Okay, so for the rest of the talk, I'm going to tell you a little bit about what we've been doing in terms of satisfying the first goal, but also laying the groundwork for the second. So what are we doing to identify some of these proteins that are actually regulated in this manner? Like I said, we're turning to the mouse model. So mouse is different from flies. They are not, at least in the lab, they're nocturnal, so they consume food at nighttime. So we did restricted feeding, and then we also collected different time points over the circadian daily, over the 24 hours. And we actually collected different organs like liver, heart. I think Nitro also got like the brain, kidney, et cetera, et cetera. And then we also looked to see if there's a rhythm in daily glycosylation of proteins in the mouse, just like what we find in flies. And we use the same approach, which is great. We also, as you can imagine, we're also doing time restricted feeding. So looking at giving them food at different times of the day, but that's a story for a different time. So just looking at regular restricted feeding when they're active, giving them food when they're active, you can see that I'm showing you down here the glycosylation rhythm for nuclear proteins globally in both the liver as well as the heart. As you can see, just like in flies, it's rhythmic. The phase is slightly different, but you can see that it's really strongly rhythmic in both of these different organs. So now we show that it's rhythmic, we can now start to actually look at what these proteins are. So the way that we try to figure that out is to use proteomics. Because these proteins are labeled with biotin, we can easily pull them down using streptavidin and actually do mass spec to see what they are. So that's exactly what we did. So we pull down using labeled extract and also unlabeled extract, and using unlabeled as sort of the background deduction. And then we figure out using mass spec what these proteins that are glycosylated are. And we find that there are a total of, in this first set of experiment, 284 glycosylated proteins, right, in the mouse liver. I'm just showing you the mouse, the liver sample. And out of that 284 proteins, about 34%, about 99 of them are actually rhythmically glycosylated And we normalize this rhythmicity with a nuclear proteomics data set by another group, which is great. We don't have to do it ourselves, even though we're doing it now. But anyway, so we find that there are a lot of proteins that are rhythmically glycosylated in the mouse liver. And this is a circular plot showing when they're actually most glycosylated. And not surprisingly, they're most glycosylated at nighttime, mostly in the beginning of the night, as well as like late in the night. And the other interesting thing that we did was we try to figure out whether there's any overlap between glycosylation and phosphorylation, right? Because that's something we're interested in. And we find that when we overlay circadian proteomics data, phosphoproteomics data with our data set, there is some overlap, but not total overlap, which is what we expected. So there is some overlap over here in the beginning of the day, but there's a lot of phosphoproteins that are phosphorylated during like the resting period of the mouse versus the more active period. And in the future, it'll be interesting to see like what are these proteins that are overlap and what are these proteins that are not overlapping in phosphorylation and glycosylation. When we did go enrichment just to see what these proteins are, not surprisingly, because we're looking at nuclear proteins, a lot of these proteins are transcription factors, chromatin remodelers. We know that a lot of chromatin proteins are actually regulated by phosphorylation, so it's not surprising that they're also regulated by olin glycosylation. And in itself, this is not very interesting, but we start looking into what are these proteins and start generating new hypotheses. And you can see that there are some interesting proteins there. For example, clock is known to be phosphorylated, of course, but now we find that it also rhythmically glycosylated as well. And you can see that this particular phosphorylation site, this one that is important for DNA binding, is actually antiphase to at least global glycosylation of that protein. And so in the future, it will be interesting to study like how that interplay regulate clock protein activity. This is another protein, XNF1 alpha, so hepatic nuclear factor 1 alpha. This is a protein that is known to actually regulate OGT. So this will give us some new ideas as to how perhaps OGT is regulated by metabolic input and the clock as a feedback mechanism. So these are only two of them, and we have now a list of proteins that we can actually dive into. And on top of that, we're working on cytoplasmic proteins as well. So just to summarize what I've told you in the last part, olin glycosylation of nuclear proteins exhibit daily rhythmicity in mice, just as what we found in flies. We identify a number of mouse liver proteins that exhibit daily rhythmicity in glycosylation, and some actually also exhibit rhythms in phosphorylation, which makes them more interesting, at least to me. And understanding the interplay between these two modifications, we think will provide functional insights into how glycosylation, nutrient-sensitive modification is actually going to perhaps remodel the phosphoproteome to regulate circadian physiology. Just to wrap up, all this work, like I've told you, even in the mouse, it's actually done by one grad student. This is other people in the lab working on all kinds of different things, including strawberry tests. Want to thank our collaborators, our mass spec collaborators in both Germany as well as in Rutgers, and also our metabolomics collaborator, the West Coast Metabolomics Center. And so this is all funded by NIH. Yep. And so I'm happy to answer questions. Yeah.
I have a couple of questions online. Let me go over the microphone, Joanna. Jean asks, great talk. Do endocrine signals play a role in integrating all the rhythmic signal sources, including light, feeding, and others throughout the body? Or does this integration happen more at the tissue or cellular level?
Yeah, no, we haven't looked at it, but I can imagine that it does. I mean, you probably can answer that question better than me. But yeah, we haven't looked at that yet. But that's why we're interested in actually looking at inter-organ signaling and how the brain actually regulates, for example, cardiovascular signal, cardiovascular rhythm. And that's something that Nitro is working on right now. Yeah. I mean, in the flies is kind of difficult, but yeah, I think, and I can imagine that in the mouse, we can probably start looking at some of these questions that you're asking. Yeah.
I have a couple of other ones, but before I jump on that, have you looked at the glucose transporters?
Not yet. No, we haven't looked at. So that's why we've been trying to optimize our labeling for cytoplasmic proteins, because we're pretty sure that a lot of these other signaling receptors are modified, but we're not totally trusting our labeling first for cytoplasmic proteins, because there's contamination from aniline glycosylation. But we're working on that. Yeah.
Okay, if you guys have any question here in the audience, please let me know. I'm just reading some of the questions from online people. So one of my students actually that is not here, Jonathan, he asked, is there any work to show that shift workers who have higher levels of diabetes, obesity, and cancer have disrupted glycosylation rhythmicity?
No, so I would say not shift workers per se, but we know that people with diabetes have disrupted glycosylation, not rhythms per se, because I don't think a lot of people have actually looked at circadian rhythms in these modifications per se, but definitely just glycosylation level in general. People with diabetes, their glycosylation level, it's different, and I would imagine that it would be, yeah.
Maybe subjected the mice to chronic jet lag. That would be a good way to compare the glycosylation on your, I don't know, liver cells. For example. In animals in 12 hours like dark cycles versus chronic jet lag.
No, I've been a fly model person for so long. I get scared when I start working on mouse, and I wanted to actually write a grant on that, but I'm like, I don't know.
Have to be.
Yeah.
Okay, there's one more question from Shamin. This might have been mentioned and I miss it. He missed it. Are there any rhythm-related phenotypes when the glycosylation genes are manipulated by mutations, overexpression, or knockdowns?
Yes. Yes, there is. So there were papers, actually back when 2013, that was the first paper when they have OGT knockout flies and RNAi, et cetera, and there's a difference in locomotor activity rhythms. And also, so we also showed that in our 2019 paper, and it's also shown by another paper in BMAL1 glycosylation by a guy in Yale, and he also showed that. So there's definitely, if you globally change OGT and OGA, the two enzymes that add on and take off glycosylation, you do get rhythm differences. Yeah.
Okay. And there's one more question in whether glycosylation or the role of glycosylation can influence the subcellular localization of the various proteins and really influence the core machinery in that way. Whether basically the question is whether trafficking of proteins can be influenced by glycosylation.
Yeah, that's a good question. So it does. It actually affects per localization. We show that it also affects DNA binding. That's why, you can almost think of glycosylation as kind of phosphorylation. And whatever that phosphorylation does, glycosylation also do, to be honest.
Good. Yeah, Silvia.
I was wondering, your mass spec data should allow you to look at any phosphorylation modification.
Yeah, so that labeling approach that we use, you can now label anything. So we're starting to label lipids, and that's another metabolic input that we're looking into. So yeah, definitely. Yeah.
Or even on the data you already have, you should be able to look at protein acetylation.
Yeah. We just have to look, but yeah.
So there's one more question. What is the typical time scale associated with glycosylation? I mean, it's like you eat, and what is that?
It's pretty fast. It's actually pretty fast. Like we haven't done like trace labeling and things like that to really know, but based on just the correlation data we have, it looks pretty fast. Yeah. I don't know how fast in terms of like seconds, minutes, or whatever, but yeah.
Yeah, so the question really says whether a fluctuation in glycosylation more reflects the activity of the enzyme.
Yeah, there's definitely delay too. You know, when you're looking at, let's say, the fly data and the mouse data, the phase of glycosylation, interestingly, the peak actually it's the same. Like even though one eat in the day, the other one eat at night. So what we're interested in also exploring between the two data sets is where is the delay? Where's the lag that actually, like where's the lag and which enzyme is actually creating sort of the lag? Yeah.
If I don't remember wrong, Sachin's data suggests that we should eat carbs in the morning and fat at night, right? So which you should be able to see more glycosylation early on and sometime during the light phase.
Yeah, you know, the
Which
lipids is interesting because
when
lipids,
first-
there's also, this is why we're starting to look at lipids, because lipids also regulate glycosylation.
Oh.
Yeah.
Yeah, sure. Yeah, you're right. I forgot my basic biochemistry. Okay, any other question? Jesse?
Yeah, I had a question about the initial fly experiment with the different feeding times. So if you take the flies that had the unnatural feeding time and then switch them immediately to a natural feeding time, do you see a restoration of their vigorosity?
That's a good question.
Yeah, it's a good question. We haven't done that, and we don't know how fast it switches back, but that would be an interesting experiment. Yeah.
So is it separate populations of flies, or is it the same population of flies?
Three different, yeah, because we have to kill them. So yeah, so it's three different populations, and this would be relevant to like shift workers, right? Do they actually, how long can they actually be in the shift work and be still kind of healthy? So that would be a relevant question. Yeah, no, that's a good question. Yeah.
Any other questions?
This is sort of like a reminder of people
Uh,
exercising
no,
while they're.
We're causing the stress on the speaker to see how the speaker responds.
Yeah, no.
Totally works. Any other questions, Shikoko?
I will ask an unsophisticated question. Do flies become metabolic disease?
Yes. Yeah. No, you can actually, it's a model for diabetes. Some people actually use that, and you can actually feed them with high fat diet. There's also genetic models. So yeah, definitely. Yeah.
So the next question is, what is like diet composition, like high fat, high sugar, high carb? What's most important for the rhythms of the glycosylation?
We haven't.
Like, how to eat.
Yeah, no, so that's interesting. So we have not tried different composition of food. We have only actually used the regular food that we feed to flies and actually move the timing. But I always get that question, like what happens if you actually have high fat diet, etc. So we have, one of my undergrad has done high fat diet and look at glycosylation is totally dampened. Yeah, and one of our other undergrad. Huh? They do, yeah. So you can actually open their abdomen up and stain them with lipid stains, and yeah. So one of my undergrad actually did that project, and then one of my other undergrad actually did like antibiotics because they want to know whether getting rid of microbiome is going to affect and it does too. It totally dampens.
Oh,
Yeah, it's very sensitive.
Brenda.
So related to this, though, I'm working with Shihoko and Evan over here on flavonoids that are known to affect rhythms in humans. A lot of other small molecules also, right? So and this is a classic picture right here. This poor kid is eating a bunch of garbage.
She doesn't look like a poor kid. She looks like a
Right, so.
very happy
But it
one.
also goes to Carla's question about, the composition. So what we're feeding these flies is really probably what this kid is eating, not what's in the background, which is what they prefer to eat.
Yes.
But it's really complicated, right? Because there's a lot of things in the diet that are impinging on our metabolism. And people, you walk through the store and there's aisles full of sodas and you're going, what's that doing to your rhythms, among other things? Right?
Yeah.
So,
But
I don't
in fact, you know, if you drink sugar, drinks with high sugar at night, right? You stay awake.
even without the caffeine.
Yeah.
right? So, yeah, so it's a big issue of also,
Longer.
how does the quality of the diet affect what's going on?
Yeah,
physiologically,
absolutely.
right?
Yeah, no, absolutely true. And then, but of course, like this would be sort of the next step. And, but then of course, like you will need, in order to actually study this, and, you want to study something with bigger differences, I guess it's easier to publish. So the first thing we did was we changed the timing, but I think all those would be doing something to the rhythms. It's just that whether you can actually monitor it, it's sensitive enough, the assay that we're doing, et cetera, et cetera. Yeah.
So the question is that if you eat the same number of calories, or if you eat the same number of calories with different compositions, right? So it's like if you have, I don't know, 2,000 calories on carbs and 2,000 calories in fat,
Mhm.
right? Something like that.
And 2,000 calories of what's behind this kid? You're probably not-
Samir, you have a question? That's what.
Is the gut microbiome having a distinct rhythm from the entire organism itself, or do they align this to a certain degree?
They're actually closely linked. There is paper from, mostly the group from Israel. They really look at just how the host metabolism and the host food consumption affects the microbiome, right? But then also, if you disrupt the microbiome, how that's affecting the host metabolism. There's sort of a bi-directional relationship. And yeah, so there's two different rhythms, but they're linked. Yeah.
Okay, we're gonna stop here because Joanna has a really busy schedule this afternoon. So for those of you that sign up for lunch, could you please wait for me outside the room? And let's thank Joanna one more time for her time.